This is a slightly longer version of a presentation I made on September 9th at the MOC3 Models of Consciousness conference. It is about the catecholaminergic neuron electron transport, or CNET, action selection mechanism. See on your screen is shows the substantia nigra pars compacta at the ganglia near the brain stem, and it just gives you an idea of how small those nuclei are. I'll be providing an overview of the CNET action selection mechanism hypothesis, some of the predictions that were made in 2018 based on that hypothesis and subsequent evidence, and then I'll provide some ideas on how to include CNET in models of consciousness, specifically the global neuronal workspace and integrated information theory. So the catecholaminergic neurons, specific interest for this presentation are the substantia nigra pars compacta, which provides dopamine to the striatum, and the locus ceruleus, which provides noradrenaline to the frontal cortex. As you can see, these are viewed as modulatory signals and not excitatory or inhibitory signals. I would also note that the locus ceruleus, or LC, is directly connected to the frontal cortex and thus has connections to the neurons of the frontal cortex, but that the SNC only connects through the striatum to the frontal cortex, which is important, as you'll see shortly. So CNET was proposed in 2018 based on this unusual concentration of large dopamine neurons and high concentration of ferritin and neuromelanin in this nuclei. The hypothesis is really a bunch of questions too, because I didn't have a whole lot of evidence in support of it, but there was some. The first is that there's ferritin and neuromelanin structures in the SNC and LC, and there's certainly some evidence of that, but not enough evidence to determine if there's sufficient ferritin and neuromelanin to transfer energy and if they have the correct physical properties to do so. They would have to provide a switching or routing function, because otherwise the energy would just go anywhere in the group of neurons, and for it to be to serve a purpose primarily related to generating action potential in a neuron or neurons, it would have to have some routing function. And looking at those neurons, it would seem like the reason for helping one or more reach action potential would be for action selection. So why action selection? Well, these large dopamine neurons are at the top of the direct pathway, which is associated with action selection. And they have these extensile axonal arbors. Now, of the million or so dopamine neurons in the brain, most of them are small. These very large dopamine neurons are maybe 1 to 2% of that total. They connect to cortical axons at striatal dendrites. So you see these dopamine varicosities meet at these boutons on the striatal dendrites, and there's cortical inputs very close, and it's a very unusual configuration, and it's these cortical inputs that have been shown through predictive reward signal studies to lead the firing of the dopamine neurons. So if these large neurons were associated with action selection, they could coordinate it. And this mechanism could prevent seizures by selecting one of these large neurons to fire instead of allowing them all to fire simultaneously, or some of them to fire simultaneously and result in a seizure. And I also know that they could integrate the cortical signals when no action is being selected. There are upwards of 1 million possible synapses in these neurons, so the actual number that are active is much smaller. But with all of these cortical inputs, there's effectively an integrating mechanism with these thousands of neurons and all of these cortical inputs through this CNET mechanism, as I'll show in a little bit more detail. Now, one of the drawbacks of the hypothesis is that kind of textbook large SNC dopamine neuron theory was that the phasic dopamine response does not code movement. And again, this was based on predictive reward signaling studies and not movement and action selection studies. But it's interesting because this paper cited this evidence in support, and this evidence is readings from a microelectrode, which was inserted into the substantia nigra pars compacta of a monkey. And the monkey was trained to see a door open and then to decide whether or not to move the arm and move the arm into a box. And as you can see, this one dopamine neuron had a lot of firing activity prior to movement, and so did this one. So, the question is, was this predictive reward signaling? Because after the door opened, the monkey saw that there was a reward greater than expected, or did that only happen after the monkey opened the box? It's hard to tell from this information. There were a lot of other neurons that did not have this kind of behavior. And again, they took hundreds of neurons with these microelectrodes, and a small number were like this. And that would be consistent with the relative percentage of the large dopamine neurons to smaller ones. But again, I think that the important point to note is that the predictive reward signaling and action selection are two different things. Action selection occurs before you get a reward and determine that it's larger than expected, and predictive reward signaling comes after that. So again, this mechanism involved all of the large SNC neurons that were connected by a Ferritin electron tunneling mechanism, and it predicted that there was a gate circuit that would sense the impedance to the next layer, the striatum, and select the path that had the best configuration for firing. And that is consistent at some level with his predictive reward signaling studies that showed that dopamine neurons are activated by rewarding events that are better than predicted, remain uninfluenced by events that are as good as predicted, and are depressed by events that are worse than predicted. So the events that are better than predicted would have more activity, raise the membrane potential, lower the impedance to ground, and that would result in the electrons being routed to that neuron and causing the action potential to be reached. It would be an assistance. I'm not going to get into the details of that mechanism within the neuron. It's rather complicated, but there's also a lot of evidence about that. So some of the evidence that arises from this hypothesis in 2018 is that there should be electron tunneling detectable in SNC tissue. Again, nobody had tested SNC tissue before 2018, so it had to be done in order to establish if there was electron tunneling. There should be disordered ferritin arrays in SNC tissue. There was some evidence, but not really as good evidence as would be necessary to show that there is this disordered ferritin arrays. There should be electron tunneling in disordered ferritin arrays, either in the tissue or outside of the tissue. It shouldn't matter because the ferritin arrays would be a physical structure. There could be some circumstances where what's surrounding them would make a difference, but as long as it's an insulator or even an ionic material, it would not prevent electron tunneling. There should also be the switching and routing in the disordered ferritin arrays to prevent the energy from just dispersing without any direction. And then there should be some post-synaptic activity of the large SNC dopamine neurons that drives action selection. And this was missing. This was contradicted by the predictive reward signaling work. So after I published my hypothesis, I looked into the cost of conductive atomic force microscopy testing, and I found a lab, EAG Labs in Sunnyvale, who was willing to do tests on tissue samples. And the tissue was 5 micron thick slices of tissue from AMS Bio, which is a tissue supply house. It was fixed human substantia nigra tissue. They placed it on a foil substrate so that we could perform the conductive atomic force microscopy testing, which requires a ground plane. And then they shipped it directly to EAG Labs. The testing that was done was with a minus 10 volt bias on the probe. The probe basically bounces along 500 times in a line and then 500 lines in a test for 250,000 current measurements. And there's a current measurement at each bounce. So these graphs you see here, there's three of them, one on the bottom right, top right, and on the left, are 250,000 points, and it's an image formed by plotting those. The minus 10 volt bias probe is essentially an electron source. So in the bottom right corner here, you see a control sample that had no ferritin or neuromelanin, and the current convention was as expected and in accordance with classical understanding of electron flow, which is that the currents were flowing into the ground plane, and that was a negative current flow convention. But in the top right-hand corner, you see a test scan with extensive positive currents. These positive currents don't make any sense according to classical theory, but they are consistent with electron tunneling. Essentially, what's happening is electrons, which are negative, are flowing into the minus 10 volt bias probe, which is negative. So there would have to be some unusual electron behavior to explain this, and tunneling could do that. This is a transient effect, and it changed, the current distribution changed over time. So this has all the hallmarks of electron tunneling. This sample in the upper right is an 80 micron square, just about. We increased the resolution down to a 2 micron square sample, and we were able to detect structures that look a lot like neuromelanin organelles and layers of ferritin outside of them. So this was the first time this kind of testing had been performed on substantial aggregate sparse compacted tissue, and the total cost is about $4,300. So it's not terribly expensive, and other people could also do these tests without too much expense. Now, prior to doing these tests, the best evidence I had of ferritin outside of neuromelanin organelles was this electron microscope image. These little black dots appear to be ferritin, but it's hard to tell with the resolution. This study used immunogold markers that were tagged to ferritin, and you can see there's a number of immunogold markers, but not an extensive number. So, this is an array of ferritin that was hard to tell. On the left-hand side, you see a diagram of what ferritin looks like. It's a 12 nanometer spherical protein structure. Inside is about an 8 nanometer core of ferrohydrite and other materials. It's a very complex structure, but it has been studied extensively and for electron tunneling, and there's a lot of evidence that electron tunneling is associated with this. This is similar to what is called a quantum dot, which I will not go into great detail on, but a quantum dot is essentially a very small structure that has an electron that can become coherent and have a coherence radius that exceeds the dimensions of the quantum dot. So the electron behaves differently than a classical electron. So subsequent to my publication of the hypothesis, this paper came out with electron spectroscopy image showing layers of iron in red outside of neuromelanin organelles. So this was better evidence that there are layers. This X-ray imaging technique shows iron in between large dopamine neuron soma in the glial cells. Hard to tell from these images, though, whether or not these are arrays. So I kept looking, and I found this interesting study of placental cells, which Dr. Michalik performed small angle neutron scattering on and measured increased magnetic ordering. So what does this mean? All right, so these ferritin layers in this placental cell got Dr. Michalik's attention, and she used small angle neutron scattering because that's a way of testing to see if there's magnetic ordering in a quantum dot solid. Quantum dot solids, again, we have these quantum dots, but they have this unusual behavior, and one of the unusual behaviors is increased magnetic ordering. And what Dr. Michalik found was that there was substantial increased magnetic ordering when she extracted the ferritin and measured it in bulk form. There was not a very good substantial magnetic ordering. So this is an indication that ferritin is behaving like a quantum dot solid. I found an electron microscope image of a glial cell from a SNC tissue. It did not identify these black dots as ferritin. but based on the striking similarity, it appears that it is, and that it has an array structure. So there is some good evidence of a potential array structure in these tissues, in addition to the conductive atomic force microscopy tests. This study was performed by Professor McCott, and he looked at small angle neutron scattering for layers of ferritin that he burned the protein core off. So it's just the protein shell, I'm sorry. So it's just the ferritin cores, the ferrihydrite cores on a silicon wafer. And he also measured this increased magnetic ordering, again, indicating that the ferritin arrangements are behaving like a quantum dot solid. And quantum dot solids, as you'll see, can conduct electrons. So in regards to tunneling through disordered ferritin arrays, after the hypothesis was published in 2018, Professor Macapetier did a study where she disposed layers of ferritin between electrodes that were 40 microns apart. The layers were formed in a random deposition. Ferritin is a nanoparticle. It's difficult to manipulate, so she used a layer-by-layer deposition process to create some structure, this multi-layer structure. And she measured 0.3 microamperes across this 40 micron gap at 3 volts. So that's a very, very long distance. Keep in mind that the soma of the SNC, large SNC neurons may be separated by as little as 5 microns. So 40 microns is a much larger distance than would be necessary. She also looked at conductive atomic force microscopy currents through the layers, and after 40 nanometers, they dropped off, and the current was in the nanoampere range. So again, this structure, this order, appears to create the ability for the electron to tunnel. That is consistent with work done by Professor Middleton, who was one of the pioneers in studying electron transport through arrays of quantum dots. And in this study, he had a disordered arrays, one layer, and increasing size, and he showed that the voltage current plots were log linear. That was consistent with the information, the data measured by Professor Macapetier. And I was inspired by this work to build a 4-terminal device instead of a 2-terminal device to look for routing. And we could also measure current across a distance. In one of my test dies, there was an 80 micron separation. We measured 3 microamperes across a 4-layer configuration at 10 volts with this kind of log linear behavior. So this is all consistent with the hypothesis of electron tunneling in disordered ferritin arrays, which had not been measured at the time when I published the hypothesis in 2018. So the devices I designed had these 3 terminals on the right and 1 terminal on the left. Professor Kai Shen at the Ningbo Institute of Materials Technology and Engineering did this fabrication and testing. He's an expert in nano films and nano devices. And so we had the ferritin layers were formed similar to Professor Macapetier's layer-by-layer process. In 75% of the dyes, we measured no current, no appreciable current between the electrodes, which shows that the ferritin by itself is not a good conductor. But in some of the dyes where we had sufficient structure, we did measure currents. And in a few of the dyes, we had some very clear indications of switching or routing function. So what you're seeing here is electrons are flowing from one side of this device to the other, but they are not flowing from the other side back. And this nonlinear switching or routing behavior was very interesting. The cost for this testing about $10,000. So again, this is test. There's additional work that should be done and can be done, and it's not terribly expensive to do. The explanation for this I determined was a Coulomb blockade. So because this is a random, the process for forming these layers was a random arrangement of ferritin cores. If there was a constriction like this, you would have a situation where the cores, which are ferrihydrite or iron in a +3 oxidation state, would receive an electron from an electrode, and it would raise the oxidation state to +2, but there would still be some +3 states. So the electrons could continue to tunnel from right to left, because there's always a more positive state, and electrons are attractive to positive. Their negatives and opposites attract. Going the other direction, though, the constriction would result in a blockade of the ferritin rising to the +2 oxidation state. And this is called a Coulomb blockade. And this has been studied in quantum dot solids, and it's been observed. Now, after I published the results of my tests, Professor van der Zandt tested ferritin at 4 degrees Kelvin and with single electrons. So you can't use single electron measurements at room temperature and ambient conditions, which is what these tests that I performed were conducted at. The single electron tests allow you to be more specific and determine exactly what's going on. And Professor van der Zandt found excellent agreement between the experimental data and Coulomb blockade theory. So this is a very good confirmation of the work that I had conducted. So going back to this predictive reward signaling of dopamine neurons being understood to be a broadcast or modulatory signal and to be affecting a maximum number of synapses in the basal ganglia. In 2021, Professor Pascal Kaiser at Harvard published this work where he stated that for movement control, dopamine modulates moment-by-moment activity in the striatum to mediate action selection. And this was based on some very in-depth work and analysis of the way that these varicosities release dopamine and how fast and how close they are to the striatal dendrites, and also how many are active. So basically, this structure would allow the conscious experience of action selection to involve the integration of these cortical signals through their effect on these strong electron interactions in the Coulomb blockade. And again, that's a lot, and I'm not really going into great detail on some of that, but this was essentially predicted and somewhat contradictory to the predictive reward signaling work, but not really, because it's action selection and not predictive reward signaling. Professor Kaiser also previously had found that the architecture supports spatially and temporally precise coding for dopamine, and that only about 20% of these varicosities are active. And this is a programmable structure. It's similar to semiconductor programmable memories and stuff like that, field programmable gate arrays. So this is a memory mechanism, most likely. The glutamate release from the corticostriatal terminals controls dopamine release, as found in the predictive reward dopamine signaling studies. And it's noted that that would apply to this action selection mechanism too. So again, if you have cortical signals coming in, stimulating these neurons, something has to help control which neuron fires to prevent multiple neurons firing and seizures. There has to be some sort of routing mechanism, and the CNET hypothesis would provide such a routing mechanism. I also note that for the locus coeruleus, similar varicosities and spatiotemporal activity has been observed. Unilateral damage to the LC and the SNC results in behavioral and memory changes, loss, and it can be resolved over time. So this indicates that those varicosities are programmable and can be programmed to essentially correct the damage or accommodate the damage. So to include CNET in a computer model of consciousness, you could go to the specific cortical neurons and the specific striatal dendritic synapses and the specific SNC neurons, which would be a lot of mapping, but this would be inputs to a neural network, and this would be used for individual action selection prediction, not populations. And there's a similar configuration that can be used for the LC, but it's a little bit more complicated. I'm not going to get into it. So a neural network has a number of inputs and outputs, and these hidden layers are connected algorithmically to adjacent nodes, and you train it with different scenarios or input samples. But for the substantial nigra parts compacted neurons, the number of inputs and outputs would be very, very large, and it would be very difficult to do this. A simpler way to do this would be to use kind of primitives. So for example, if you wanted to model driving on a remote two-lane highway, you could have a group of primitives for speed limit, for distance to the car in front, and so forth. And this would be cognitive process signals that are coming in, stimulating the striatal dendrites. And then the outputs would be the selected striatal dendrite or dendrites that cause an action selection to occur, or no action selection, because no action is a decision also. So this could possibly be used to model a conscious state, the conscious state of driving down the road at the speed limit, knowing that the car in front of you is far enough away, there's no obstacles. All of a sudden that car slams on the brakes, so you press the brakes, or you turn left or right. This would be a moment by moment modeling of the conscious state that could be used for some purposes in modeling consciousness. Now, the global neuronal workspace models model neurons as neural networks, not artificial neural networks, but you could hook them up to an artificial neural network with a sufficient level of granularity. I'm not sure that they have the granularity necessary to provide inputs to the striatal dendrites. Right now, at least in this more recent paper, dopamine is modeled as modulatory down in the cognitive level, as opposed to the conscious level. So there would need to be some modification of GNW to model this mechanism that's been discovered by Professor Kaiser. This model was a GNW inspired computer game character control. It had the workspace modules, but it had action selection and behavior modules as inputs to the workspace module instead of building the action selection into the workspace modules, which would be more accurate. For CNET, CNET uses this transition probability matrix and digital models of neurons. So for IIT, I'm sorry, uses this. IIT could be modified to model CNET. CNET is not digital, so it's a function of dopamine levels, which generate the electrons, impedances, which determine how the electrons are going to flow, the ferritin connections between these neurons. So all of that would have to be modeled, and it would be an analog and not a digital type model. But again, this could be accommodated, and it could be hooked up to the digital mechanisms that reflect cognitive processing, as opposed to this integrating mechanism that it could be responsible for consciousness in some manner. So again, the another way of looking at it is that you have this shared latent space that's been hypothesized, or the neural correlates of consciousness, which is where the SNC and LC would sit. The cortex would be a pre-conscious experience, and the remainder of the striatal cortical loops would be the post conscious experience processing. But all of this would not be part of the conscious experience, which would only be in the neurons that are associated with the integration of the electrons. This is probably a better way to look at it, where you have the soma of the neurons and connections with ferritin depending on the activity level. Glial cells have active connections between neurons, or they're passive. So you have routing of electrons, and this would create some kind of mechanism that could possibly represent various states. It's a high frequency analog electron flow. The electrons are interacting in strong electron interactions, and are formed by quasi-particles. For example, an electron in a hole, or an exciton is a quasi-particle. There are magnons and phonons. It's a very complicated structure, and it would require a substantial amount of additional analysis to determine how it functions. So in summary, in 2018, I made a hypothesis, subsequently did some testing, and monitored work by others that has provided evidence that was predicted by the hypothesis and is consistent with the hypothesis. And this mechanism could be included in mathematical models of conscious. It's not itself a consciousness model, it's a action selection mechanism in a substrate of consciousness. And again, I do have to thank people who have helped me. I'm a citizen scientist and do appreciate any help I can get. So thank you very much for your time and for listening.